All right, let's go ahead and get started. So thank you everyone for joining our workshop today, which will be on expansion microscopy using Magnify. I'm really excited to have our speaker here today, Dr. Yongxin Zhao, who's an associate professor at Carnegie Mellon University. I am Afrida Rahman Enyer. I am a scientific liaison and product manager at Proteintech, and I will be your host for today. Before I pass it off to Dr. Zhao, I just wanted to share a couple company slides to introduce Proteintech to you. So Proteintech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass many different research areas and cover many different targets. We also offer ELISA kits, IHC kits, antibody labeling kits, and magnetic cell separation kits. Within our Proteintech brand, we also have a few other lines. So we have our humankind branded cytokines and growth factors. So all our cytokines and growth factors are expressed in human HEC293 cells so that you can be confident that they have native folding, post-translational modifications, and glycosylation that you would see in human proteins. We also have our Chromatech branded nanobody-based reagents, and nanobodies are essentially really tiny antibodies, so they're about 10 times smaller than traditional antibodies. And because they're so small, they can get really close to your protein of interest, so they're great for things like immunofluorescence and super resolution microscopy. They also have very high affinity for their target, so they're really good for clean immunoprecipitations as well as protein purification. Our motto as a company has always been that we are a company for scientists by scientists. Our high quality and award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. We also know the importance of product validation and generating very clean antibodies. So, whenever possible, we do try to validate our antibodies using knockout, knockdown models. And then also as antibody experts, we have in-depth knowledge in immunoassay applications, and we can offer outstanding PhD level technical support. So with that, I want to go ahead and introduce our speaker for today, Dr. Yongxin Zhao. So Dr. Zhao is an interdisciplinary scientist and engineer with notable contributions and pioneering work across protein engineering, neuroscience, chemistry, and optics. Dr. Zhao's recent endeavors focus on advancing expansion microscopy, developing novel imaging techniques that enable nanoscale visualization of biological specimens. His contributions include enhancements to expansion microscopy that permit detailed imaging of clinical biopsies and complex biological systems, resulting in several high-impact publications in journals such as Nature Biotechnology and Science. As the leader of the Biophotonics Research Lab at Carnegie Mellon University, Dr. Zhao's work is dedicated to inventing and refining technologies that improve our understanding of the brain and other biological entities at a molecular level. His current projects also extend to developing computational tools, including sophisticated deep learning algorithms to analyze nanoscale imaging data. These technological advancements hold promise for pioneering new diagnostic approaches and are part of Dr. Zhao's broader mission to disseminate these cutting-edge imaging tools to the global scientific community. So with that, I would like to go ahead and pass it off to Leon. Thank you so much for joining us today and for sharing your work and all your tips with us.
Okay, sounds good. Thank you so much for the introduction, Rita, and I really appreciate that. And I'm going to take over. So I believe you also now can see my screen. And so I would say good morning, good afternoon, good evening, depending on where you are in the time zone. Thank you so much for coming to this webinar, and it's my great pleasure to be here to share some of the research story that we have on developing this improved version of expansion microscopy using Magnify. So I'm going to outline some of the agenda for this particular webinar. So first, I will give you a little bit introduction about what expansion microscopy is all about. And then I will talk about the latest version that we developed in the lab, Magnify and microMagnify, and each of them will supplement with some case studies. And then I will address a little bit about if you want to do some Magnify expansion microscopy, like what kind of resources that you will have. And then I will answer some of the common questions related to expansion microscopy. And then, of course, we will have this Q&A at the end. So let's get to the biological system. If you take a look at them, we will actually realize they are incredibly complex machines that are basically assembly of all kinds of different biomolecule, protein, nucleic acids, and metabolite, all assembled together and orchestra at nanoscale precision. Even though the scale of the size of these like biological system actually span a wide range, right? We can see, like, for example, like human organs is like tens of centimeters in size, all the way down to virus. It's like a couple, even like tens of nanometers, and down to the biomolecules, like 1 nanometers. It spans for several order of magnitude in size. In order to study those things, we need tools. We rely on microscope to visualize some of those processes. Because you may notice that it works on, depending on the type of microscope you chose, it works for different range, right? For example, electron microscope, you can see very tiny details, like those protein levels, for example. However, it has hard time to see, like, anything larger than, in the millimeter range. And then the light microscope actually is more common in our research lab, and it can image all the way from roughly 300 nanometers all the way to centimeter range. However, there's a barrier, as you can see. You cannot actually get into the nanoscale, right, with a conventional light microscope. Why would that be, right? The reason is that light microscope use visible light. So if you try to, for example, turn to some of the very small structure, let's say synapse. Those are very small, tiny units, which is very critical for computation of the brain. And if you try to zoom in, you will see something like this, right? Using the diffraction-limited confocal microscope, for example, it's a pixelated, meaningless patches. The reason is that you use the visible light, and which is around 350 nanometers all the way to 700 nanometers in wavelength, and the object, the synapse, are pretty close to that range. Therefore, if you shoot light there, the diffraction will happen. So you will not be able to really focus on this. And so you will see this blurry images, no matter how hard you try to zoom. Of course, like, people try very hard to circumvent this so-called optical diffraction limits, and therefore, invent this a bunch of super resolution microscopy methods. And with that, you will be able to see the details, right? So here shows you like 2 different colors. One represents the presynaptic markers. Another one shows the postsynaptic markers. And you can see very well, you can pinpoint the location of the proteins. The problem is that it pays a high price, right? To get the information down to the nanoscale, you always need to pay a high cost. Like, if you recall the electron microscope, it's not a cheap piece of equipment. Same thing for the super resolution microscope. It's not only expensive, but also slow, right? It's slow, expensive, and complex. You have a steep learning curve to learn it. So it's not very easy to use this to scale your biological insight if you want to do some discovery and at large scale. So we need some out-of-box idea. Let's say if our Earth is the targeted species, right, and as specimen, and then we try to use diffraction-limited microscope to image, let's say, some of the cells. Let's say Mellon Institute, that's where I'm working. You will see something like on the left panel, right? So you see a lot of cells. You locate the Mellon Institute, but you cannot see the details because the resolution limits. However, if you think about it, if you can actually enlarge the Earth several times in each dimension, imagine everything inside the Earth actually also enlarged. In this case, using the same microscope, you will be able to see the enlarged building of the Mellon Institute, and therefore you'll be able to see more detail. For example, I can see those air conditioning units on the roof, as well as other equipment, as well as the cars on the road and the trees. So you can see a lot of detail. This concept is called expansion microscopy. And it has been around for a while now. The first paper published in Science by Professor Ed Boyden's lab at MIT, that's also where I had my postdoc training on it. Since then, it has been almost a decade, and many different versions of expansion microscopy has been developed. And I was fortunate enough to be among one of the first wave of inventors, in this field, so be able to contribute to this. But basically, even though there are many different types of variants, you can always boil down to several steps. So here I'm going to just summarize this here. But this is the schematic. So always first you have to apply a small chemicals to modify the target type of biomolecule of interest that you have. For example, you want protein or you want nucleic acids, you will have to change the chemicals. But basically, what the purpose is to modify the biomolecule so that you install a handle over there. The handle will be able to incorporate the biomolecule and covalently link them to the hydrogel network that you're going to generate in the next step. So once you modify the biomolecule, the next step is to infuse the specimen with the building blocks of hydrogel that can be commonly found in, for example, baby diaper material. So, for example, if you remember baby diaper, you add water, they will swell, right? So we kind of use that kind of property to enlarge or inflate the specimen, if you will. And there are different types of composition for this kind of hydrogel. I'm just showing here, depending on how large you want to expand them. The typical one is the sodium acrylate and acrylamide-based polymer, as shown right here. And also you can add some DNA and then change the composition, and you might be able to expand even bigger. But the deal is you infuse the building blocks of those hydrogel into the specimen. They will penetrate very deeply because they are very small. After that, you will trigger so-called in situ polymerization. Basically, what happens is those components will cross-link to each other and form a gigantic three-dimensional hydrogel. And during this process, the modified biomolecule will actually covalently link to the hydrogel. And after that, you have to go through the homogenization step. Basically, you need to apply either enzyme or apply heat treatment to denature the protein or digest the protein so that the protein, the structural protein exists inside the tissue would no longer get in the way, right? So those interaction already cancel out or eliminated. What I mean is after the digestion or heat denaturation, the only mechanic support will be provided by the hydrogel. And then the spatial location, remember those targeted biomolecules actually covalently link to the network. The hydrogel network can serve as a coordinate, a 3D coordinate. Therefore, if you add pure water, this hydrogel will actually swell, expand homogeneously in 3 dimensions because there's no structural protein get in the way. And the biomolecule that decorates the hydrogel will actually also isotropically separate from each other, while still maintain the overall shape and the structure that way. So therefore, you will get this expanded sample. And then you, of course, if there's labels there, then you will be able to see those labels using microscope. And here just shows you one of the example on the right side. There's a mouse brain slide that embedded in the hydrogel that I will just discuss. And then after you homogenize and then you add the water, they just swell. This one swells 4.5 times. And then you can see that it becomes transparent because essentially expansion is a dilution process, right? So it becomes much more diluted. Therefore, you will not be able to see like those scattering effect caused by the tissue proteins, for example. Now, next slide, I'm going to talk a little bit more about anchoring. So we have been developing many different versions of expansion microscopy, relying on different kinds of anchoring reagent. So that means that there was no really a one-stop shop. you have to really shop around depending on what kind of biomolecule you want to choose in order to choose the right anchoring methods. For instance, like at the beginning, like the very first generation of expansion microscopy that published in 2015 rely on customized conjugate complex to recognize the target that you want to image. It's really complicated. Of course, I'm not going to dive into the detail, but basically it's a DNA, functionalized DNA conjugated antibodies. And then later we found that there's a small molecule called agars that can actually modify the proteins directly. And this agars has a small handle shown as the blue that can incorporate into the hydrogel. So that makes everything much more convenient. And there's other similar chemical has been published. And also, for example, like this MA-NHS as well as MAP. So these are for the proteins. And then for nucleic acid, you have to use a different chemical. So there's a label X that can incorporate the nucleic acids into the hydrogel. There's also like lipid anchoring reagents. And of course, by no means, this is not a comprehensive review of all the anchoring methods. So there are others around in the literature. But the point is, there's no one-stop shop, right? There's no any universal anchoring mechanism before Magnify. Then another thing, other limitation, right? Even though there's many different types of expansion methods, majority of them are still expand only 4 times, which are not quite sufficient because it's only provide you about 17 nanometers resolution, depending on what kind of objective you use. That cannot resolve many useful ultrastructures. For example, like the synapse ultrastructure. And they also have limitation in terms of like samples, type, capability. Most of the expansion microscopy method use on brain tissue because it's very soft and easy to expand, as well as culture cell because it doesn't contain the extracellular matrix. And of course, like many of them use proteinase K to digest the epitopes or the structural protein. I mean, I guess the epitope is kind of the victim of this method, right? Because you digest everything, it's non-specific. That means that the epitope loss. So you cannot actually stain the sample after expansion. You have to do the pre-expansion staining. You need to make sure everything's stained, and then you apply the expansion microscopy methods. And also, many expansion methods requires several days process to do. And because the gel is quite delicate, it does require some skill, in order to handle this properly. Otherwise, it will keep breaking the gel or suck them into the pipette tips. So we try to address this issue and make it more easier to use, lower the barrier for users. That's one of our missions. So we developed this Magnify, right? It contains all these features that are improve compared to the traditional methods. For example, like universal anchoring, it can expand up to 11 times, and then combined with some other method, you can actually get to 15 nanometers resolution. You can expand different kinds of tissue with different kinds of formats. And then we also preserve the epitope because we don't use enzyme anymore. We use a heat treatment to denature protein, so the actual protein are still there inside the hydrogel, and then you will be able to apply antibodies after you gel it and then homogenize the samples. And then most beautiful is that the Magnify can actually can be done within a day. And it's quite easy to do. So actually even a non-expert will be able to finish the process within a day with a high success rate. Each of these I will talk about a little bit case study. So this is actually the workflow. As you notice, that Magnify doesn't have the anchoring step. So all you need to do is actually just infiltrate the gelling components inside the specimen, because the gelling component contains the methylcholine, which is the key component that can incorporate all the biomolecules, not just protein, but also nucleic acid, but lipids and carbohydrate, so on. So that happens during the gelation process. So you don't need actually anchoring in this particular case. And after that, of course, you need to do heat denaturation treatment. And after that, you wash properly, and then you can do the staining to use your favorite antibodies and stain your target. After that, you expand, and then you can image. Now, we first talk about universal anchoring, right? So we do have data support that Magnify indeed incorporate the protein and actually incorporate protein in higher efficiency compared to the agar-based protein retention expansion microscopy. And this actually conclusion can last across different types of tissue, right? Kidney, brain, lung organoid. And then, of course, we also see that they can incorporate nucleic acid, for example, DNA, RNA. This is just one example. We try to simultaneously stain DNA, which is TAPI, RNA. We target the GAPDH as well as the DNA fish, which is the purple one. And then, surprisingly, we also found that the mesoclonal can also incorporate lipids inside the gel, so that you actually, even though after harsh heat treatment, you still be able to get the lipid stain. So as you can see here, you can actually use the conventional lipid stain, for example, DID, DII, to stain the gel, and then you will get that. This is one of the examples. This is a presynaptic compartment, the whole thing. Here, the yellow is the synaptic vesicle markers. We use a Proteintech synaptophysin antibodies. It's a rapid polyclonal antibody, very robust. And then we also use this conventional lipid dye to stain. So this purple little guy over here, this is the mitochondria. You can see the crystal structure, actually, thanks to the 11-fold expansion. Now, I mentioned it's 11-fold expansion, so that means that you can do many useful things, right, to see those ultrastructure, because you can actually get to very low, very high resolution. So I'm just showing you, this is mouse brain embedded in the Magnify gel and expand 11 times. On the right side, because the protein are contained in the hydrogel, therefore you can actually apply a specialized dye called NHS ester fluorescent dye. This dye very specifically, actually they only look for the primary amine groups. So they will actually label all the primary amine groups. Therefore, because lysine are pretty abundant in the proteins, right, in our biological system, so you can actually label almost all the proteins inside the biological system. And because of that, you can actually generate electron microscope fueling contrast. So here, panel 1, 2, 3 are actually magnified images, but then we actually use the pseudo color to mimic the electron microscope contrast. And you can see that this is the presynaptic dense projection right here, right? You see this pretty classic structure can be reviewed by Magnify. And compared to the electron microscope, those features are pretty similar. And because Magnify doesn't require ultra-thin sectioning, so you can actually do 3D, right? If you have a confocal, you can actually easily change the Z brain and get those 3D stack. Therefore, you might be able to find this hexagonal structure, which are also reported in the literature for this particular presynaptic dense projection, right? You can rotate, with different orientation, you will see this hexagonal structure. And we can also turn to other ultrastructure. For example, you can actually image the serial machinery, right? We know that this is a very tiny structure, right? It's about 200 nanometers in diameters, and it has a 9-fold duplex microtubule, right, surrounding the center singlet microtubule. So you can easily see this with Magnify. Of course, the resolution is not quite enough to see those dining, the two double rings inside, but you can easily count the numbers of the microtubule on the outer side. And one of the advantages of Magnify is that you can actually generate a 3D stack. For example, here just shows you one example. It's a very small section of a serial machine, right? But then you can actually span the whole C stack and be able to image the whole serial machine, right? All the way from the basement membrane, transition region, all the way to the serial, axiosome, right? So this is quite useful, which is something that are not easily done with electron microscope. That will probably take weeks in order to assemble images like this. And of course, we want to see, this is a case study. We want to see whether we can actually use Magnify to detect the defects in the cilia, right? So we call this ciliopathy. We want to see if we can detect that, whether there's enough in resolution. So we try to do a comparison, right? So this is just some classic EM images of cilia. Like this is normal-looking cilia, and then there's others that are abnormal. We try to induce those defects by introducing the drug, as well as try to get those cilia sample from like the identity that with genetic mutations. So as you can see here, this is corresponding Magnify images on the top. By counting the numbers of microtubule, you'll be able to tell which one are normal looking, which one are abnormal looking. So you still be able to sort them into different buckets. And this is one of the foundation that we use to do the following experiment. So now we try to prepare 3 different types of sample. We have the human stem cell divided lung organoid from different sources. So we have normal, and then we have a normal organoid treated with a cancer drug. It's a Taxol with a 20 micromolar concentration. And then we also have a so-called PCD group, which basically is the same type of lung organoid, but derived from patient that with this primary ciliary dyneschenia. So we have these 3 different groups. We first use electron microscope to characterize, to see a bunch of different cilia, and then try to sort them into different buckets, right? So the abnormal one, we show them in the orange, and then the normal one, show them in the blue. And as you can see, these 3 different groups has a different population or composition of cilia. Some of them has more normal cilia, just like the normal one, and then the PCD group has a majority of them are normal looking. And then we do the same thing using Magnify. As you can see, that the population of the composition that quantified by Magnify looks the same as electron microscope. There's no statistic difference, you know, when you compare normal versus normal, like Magnify is normal versus EM's normal. They are the same. Same thing for other 2 different types. So this suggests that the Magnify approach already can accurately quantify those pathological changes in a manner that's similar to electron microscope. And of course, like one of the selling point of Magnify is that it can actually expand a wide range of sample in different formats. So not only you can expand culture cell, you can expand fresh preserved tissue, FFPE sample that can be commonly found in the hospitals. So we're going to showcase 2 of the example here. First, the general characterization. We try to expand different types of FFPE sample from different organs, for example, colon, breast, uterus, like you name it. We can expand them quite easily. And then you can and then stain them with different kinds of antibody that you like. And we also try to characterize, and then we show that the data shows that the Magnify has a low distortion rate after expansion. Basically, you still see what you want to see, you know, without suspect like there's some distortion happening. That's very useful because that means that you can actually do something interesting. For example, you can work on kidney pathology. You can actually use Magnify to examine the kidney ultrastructure, just like the way that you use a clinical electron microscope to do. So here shows you, again, EM fake color Magnify images of a kidney glomeruli. And then we pick one of the region and zoom in. Now here you show the black and white is the pan-protein stain, the NH ester fluorescent dye that I mentioned. So it's staining all the proteins. And then we also have the antibody that stain the marker that you like. So you can actually see the distribution of those proteins in the context of those kidney ultrastructure. This is something that's very difficult to do with clinical electron microscope because they only have 1 colors. Now, what I mean is that here you have the sufficient resolution. You will be able to see the prototype processes, like showing here, like this finger-like structure, which pathologists typically will look at, as well as this black band, which is the glomerular basement membrane that pathology, you usually have to measure it. The normal should be around 300 to 320 nanometers. Anything thinner or anything thicker than that range suggests a disease in the kidney. So I hope that I can show you here is that the Magnify images already provide sufficient resolution to to examine this kind of pathological related ultrastructure changes without the need of a clinical electron microscope. Another difficult how to expand specimen we're going to talk about is infected tissue. Infected tissue contains pathogen. Pathogen, I'm talking about bacteria or fungus, and they contains cell walls. And cell walls not only contains proteins, but they also have the glycans and other carbohydrates that are cross-linked to each other. So traditional expansion microscopy has hard time to expand them. The reason is that proteinase K cannot effectively digest cell walls. So here we develop microMagnify. It's based on Magnify approach. On top of that, we have a cocktail of cell wall digesting enzyme. So we combine these two approach, we can actually expand infected tissue or even a pathogen or biofilm itself. And here I'm just going to showcase ones. I mean, for the other example, feel free to check the papers that we publish in Advanced Science. So this is actually one of the most difficult sample to expand. It's a human cornea. It's one of the components in our eye that contains abundant collagen. Very, very tough indeed, because it serves as the protective layers. And here we got the sample from the eye hospital, Wills Eye Hospital in Philadelphia. They provide us the infected sample, and the pathogen are very different, right? They have fungus, they have mycobacteria, and also amoeba, and those are parasites. So we successfully use a microMagnify to expand them, not only expand them, but be able to highlight or label those pathogen, and then be able to examine the morphology of them and tell which one is which, right? So for example, we can see the fungus, we can see the mycobacteria, as well as the parasite. So the last two examples that we're going to talk about, because we preserve the epitopes in the hydrogel. So that means that if you apply a set of antibody, you perform a multicolor imaging. After that, you can actually remove your antibody because the epitopes still preserved in the gel. So you can then apply a second round of antibody, which target different markers, and then you repeat this process many, many times. You can actually generate a multiplex fluorescent images that contains more than 4 colors. This could be very useful. I'm going to explain a little bit why, compared to the traditional spatial biology technique. So first, just show the example. So this is a culture cell that infected with Staphylococcus aureus. And then we also apply some Staphylococcus aureus toxin and try to change the cells. So we have a mutant, which has no toxicity, and then we have the wild type. Now you can see that thanks to the Magnify-based approach, we can actually label 10 different colors or 10 different markers. Now we can actually, with that, we can do something interesting. So traditionally, we know that there are many spatial biology methods that can label more than 4 colors. And sometimes you can get up to 100 colors, which is great. The thing is that if you try to do some co-localization analysis, you try to see how close the protein of interest they are. They actually have a hard time. The reason is that the imaging system used by those spatial biology techniques are diffraction limited. So, for example, here, you try to examine this protein called LRTAF, and then you try to see how they interact with CD63. All you see is these blurry images that they seem to overlap with each other. I mean, this is like a heat map that highlight all the interaction between all the other proteins, right? So because we have 10, so this is like a 10 times 10 matrix to show this colocalization index. And you can see that most of them are kind of overlap with each other, seems, because the low resolution that provided by the conventional microscope. So of course, with this, if you try to do the study, because we have 2 conditions, right? So we try to do study. You will conclude that there's no interaction changes between the LITAF with the CD63 and the RTAF with another protein that we care, which is NADD4. So no change. However, the story will look very different if you use the microMagnify. So what happened is now the resolution improved dramatically, right? You have a 10 times more resolution. Now the pixel size become pretty close to the protein. So the protein size is roughly from 2 nanometers to 10 nanometers. And the pixel size for Magnify can be as low as 10 to 20 nanometers per pixel. So what I'm saying is that if you see 2 pixels, 2 different colors, they overlap with each other, that means that these 2 different species, the markers, they are actually scrambled into a very small cube, right? That might be only like 15 times 15 times 15 nanometers. So that means that the likelihood that these 2 markers, they interact are much more likely. So if you look at this interaction map, you see that it's much cleaner, right? Because there's not so many like interaction, right, between a given set of proteins. So the interaction, the colocalization index here actually becomes more believable. And then we, of course, acquire the heat map of this interaction in different conditions. And now if you look back to the interaction between the LITF with CD64 as well as the LITF with NADD4, and you can see that now they do detect the changes in terms of the protein-protein interaction for these 2 particular pairs. So that suggests that there's some mechanism, right, behind how the cell respond to the treatment. And of course, I talk about all these features. Those are beautiful, like this Magnify. That's great. But at the end of the day, I mean, we hope that Magnify could be used by any research lab, just like you, right? I hope that would be our mission, so that we simplify the design of the Magnify and can be done in a day. So I'm going to show you one example, right? So this is a lab that never touched expansion microscopy, has 0 experience, only has a wet lab. So we actually send them a kit. So we actually develop a simplified version of Magnify in the form of a kit by our spin-off company, Magnify Bioscience. And then we just send the kit to this lab. And it's an optical lab. So they build a microscope themselves. It's a large wide view fluorescent microscope that can see very large area. However, as you can see, the wide field fluorescent microscope has a very poor XL resolution, right, due to the nature of the mechanism. So here, Magnify helped them to improve the optical sectioning, so they actually can generate meaningful 3-dimensional structure of these mouse colon samples, just like here I show you, right? With the help of Magnify, they can even tell the individual nuclei using a no magnification objective compared to the case without Magnify. So I hope that you will be interested in using Magnify protocols. There are 2 ways to do it. You can do it yourself. In this case, we provide you some resources. For example, we have a website that you can use to purchase those reagents. You can also look at the video protocol paper that we publish in JOVE. We also, of course, I have the Magnify and microMagnify paper. They are fully open access. So you can look at the method section and try to repeat the experiment. And on the other hand, we have a startup, as I mentioned. You can try to use the easy-to-use kit provided by those startup, Magnify Bioscience. They beta testing the Magnify kits with menu as well as detailed video protocols. Currently, it offers 4 different types that target culture cell, FFPE tissue, freshly preserved tissue, as well as organized. So if you're interested, feel free to take photos on this QR code. It will lead you to the Magnify Bioscience website, and then you can inquire the detail for the beta testing program. Now, I'm going to address some of the common questions before we actually jump into the Q&A. This is the question that are repeatedly asked by the potential expansion microscopy users. So what the first question is like, in terms of like what kind of advice, on antibody staining, right? What's the best practice to stain before and after expansion? So for the pre-expansion staining, it depends on. So the first primary antibody stain, I would say just follow your best protocol for the particular tissue that you are targeting. You can just treat it as a regular immunofluorescence staining for the primary part. However, for the secondary antibody staining, the choice of the conjugating fluorophore matters a lot. In general, highly photostable fluorophore will do much better for this type of approach. If you want to stain the sample before you expand them. The thing is that when the expansion process evolves the oxidative reagent that will breach the fluorophore. And also the proteinase K digestion version will further, weaken the signal as well, because it will digest some part of the antibodies. So therefore, it's highly desirable for you to pick the photostable fluorophore to do the job. For example, like those Alexa fluorophore AA, CF dyes, like those are really good examples. Now, when it comes to the post-expansion immunostaining enabled by, for example, Magnify, you don't have that worry, right? Actually, because the epitopes are preserved in the hydrogel, so you can just apply the antibodies. You don't need to worry about being breached by the gelling process. So, which is great. The only thing you need to worry is that you have to make sure that it's washed properly, because it's a heat denaturation process. It does involve loss of surfactant for the homogenization. So you have to carefully wash away all those surfactants. So you have to make sure that, can you still hear me? Are you still with me? Okay, so that's why you have to make sure that you wash the sample properly before you enter the immunostaining stage. And here, 1x PBS or 2x SSC with 0.1% twin 20 or C12E10 would be great. It works for most of the antibodies because the epitopes are actually separate from each other, so they no longer being masked by the surrounding proteins in the expanded state. So you don't need the blocking reagent. Actually, you can just use this this buffer that I mentioned to do the immunostaining. And of course, like the concentration ranging from 1 microgram per mil to 10 microgram per mil, depending on the abundance of epitope. For thicker tissue, you probably need to consider higher concentration of antibodies due to the much higher accessibility for the epitopes. And then another question is like, how stable is the expansion is once they perform, and also like, does the sample shrink back to its original size over time? So the answer is that for Magnify gel, for example, you can store them in a 1x PBS as long as it's supplement with some azide to prevent the growth of bacteria. They will stay for a long time, at least 3 months in the fridge. Of course, right, the size also shrink a little bit, right? It will be around 3 to 4x expanded. And as long as the gel are hydrated, you don't need to worry about they shrink back to the original size. And final common question, does the expansion affect epitope availability and as well as like the protein structure? Well, first thing I would like to clarify is that Magnify works with preserved specimens. What I mean is those protein structure has been altered by the fixation already, right? You fix them with PFA or you fix them with co-methanol, like those methods already do something on the protein structures. So we actually have a different protein structure to begin with already. If you use a proteinase K to digest the sample, of course, like you lose all the epitopes basically, right? It's very low after homogenization. So in that case, it's out of question. For Magnify, it largely preserves the epitope availability. Just like any conventional immunofluorescent, though, you do need to verify the antibody that you try to use. So you do need to do some sort of screening on the Magnify species. So we are actually, our lab are kind of a customer, Proteintech, and we have been quite successful using Proteintech's antibody on the Magnify species. For example, I'm just summarize some of the antibody that we try and works right here, right? So it ranges from like those synaptic proteins, for the neuroscience, and all the way to like the cancers. So I think we have a good success rate using the Proteintech antibodies. One recommendation would be that you try to look for polyclonal antibodies, as well as the antibody that are specifically designed for Western blot. Those, we have a better luck with those antibodies. Those are just some of the tips. And finally, this is the Q&A. So I hope that we still have time, and I'm happy to answer any question that you might have.
Great. Thank you so much for that wonderful talk. All your data was just so amazing to look at. And, being someone with a neuroscience background, I really appreciated your presynaptic densities work, and made me feel like everything I've done has been very archaic to see kind of your next gen type of applications and techniques. So thank you so much for sharing that. I also, thank you so much for doing the kind of Q&A and frequently asked questions at the end, because you addressed some of the questions that we already had in our chat. So that's great. But we do have a few others that we have some time for. So the first question is, which types of samples lend themselves well to Magnify? Which properties are going to make them more challenging?
So I think mainly is the collagen content of the tissues. But I would say that pretty much we test all kinds of different tissue type, like all the way from culture cell to muscles even. So we can actually expand them all. There's no issue for those. I guess the most hard to do sample will be actually that contains the bone components, right? Because you cannot expand the bone.
That makes sense. And have you tried in kind of like, you mentioned that it works particularly well in brain tissue. Have you tried diseased brain tissues where you see like extensive neuronal loss or larger ventricles? So like, is there an instance where the tissue just doesn't have enough integrity for expansion microscopy?
Actually, that would not be a concern. Actually, we do expand, for example, a human brain with Alzheimer's without any problems. And the thing is that at the very first step, you have to infiltrate the tissue with the gelling components. Once the gelling process are finished, everything are locked. So they don't even have a chance to disintegrate. And then after that, you expand it. So then you basically get all the structure preserved in that way. So you can see details. I mean, of course, I did not show the data here, but we do have nice images, of like nanoscale plaque, for example, like those amyloid plaque in the Alzheimer's brain.
Great, thank you. Our next question is, could this technology be combined with spatial transcriptomics?
Yeah, that's actually something that we're working on. In theory, right now, you already can do that, as I demonstrated by the microMagnify. Of course, you can also use Magnify to do. I mean, you can stain, different kinds of antibodies. But we are working very hard, try to validate as many antibodies as possible so that you will be able to use those antibodies, right, without asking a question. That's number one. Number two is we try to generate a new format that are more friendly to the existing commercial platform. Please stay tuned on those. Hopefully, like they will come out this year or next year. In the form of papers. They will allow you to do this spatial biology with nanoscale much easier.
Great, thanks. The next question that we have is asking, would it make any difference if you immunostained before or after expansion?
Yeah, I was intending to put a slide to show that. So, I mean, first thing is the signal to noise, right? Because for the post-expansion staining, of course, like you avoid the breaching of the fluorophore, therefore you will have a better, brighter signals. Not only that, before expansion, because all the epitope are crowded to each other, right? All the protein are crumbed to each other, so the accessibility of the antibody are limited. The declouding effect, thanks to the expansion, actually helps for each epitope to be bound by more antibodies if you chose polyclonal antibodies. Therefore, that gives another boost in terms of the signal to noise. So actually, in terms of labeling density, you will look much smoother if your post-expansion immunostaining are successful.
Okay, great, thanks. So overall, generally, you would recommend doing post-expansion staining if possible.
Yeah, if the antibody are validated and then works, and then I would recommend do the post expansion. And actually, in the paper, we also mentioned another aspect. It's the lineage. Think about antibody has a size. It's actually quite big compared to, for example, tubulin structure. So if you try to do pre-expansion measure, so you stain pre-expansion and then try to measure the tubulin width, you'll be like 80 nanometers or even larger than that, maybe 100. That's not accurate. However, if you expand the sample first and then you stain with antibodies, because the sample become bigger relative to the size of antibodies, so actually the size of antibody become irrelevant. We measure the width of the single microtubule. It's very, very close to the electron microscope value. It's like 46 nanometers.
Great, thank you. So we have a question about more on the, after you've done the staining and the expansion and everything, how is the sample prepared for microscopy? So, can you cover slip with such thick sections?
So there are, of course, different ways, like has been reported in the literature. Right now, there's some so-called one well plate being sold by some of the vendors. So it's a very big plate, can fit in the multi-well plate size, but with only one well. And then you can actually put your sample on top, on it with the right orientation, facing down, for example, if you use an inverted microscope. And then you can also, and then apply some agar to fix the sample in place. Those are also reported already, like in the literature. You will be able to generate relatively immobile sample for you to do the imaging for long term. And of course, as I mentioned, we try to disseminate a kit, and the kit also contains those instructions. It's actually kind of convenient for you to figure out which orientation of the sample you should put on those plates.
And then can you use kind of the traditional anti-fade or those types of methods as well?
I think in that particular case, we are trying to work on some of the imaging buffer that are anti-photo bleaching. The traditional slow fade, unfortunately, because it contains glycerol, the osmotic pressure will actually shrink the gel. So we cannot use that right now at the moment, but we will report you once we found optimized recipe on that. But I mean, overall, since we picked the photostable fluorophore, right? So actually the imaging are quite stable. Even you use a high laser power.
Great. So I think we have enough time for one more question before we do some outro slides. So the last question is, how does this technique handle low abundance proteins?
So for low abundance protein, I think we do need some amplification mechanism. Fortunately, like Magnify are compatible with some of the classic amplification approach. For example, like branch amplification. For example, like there's one other antibody-based amplification technique that we use actually quite often. I mean, like you can try to choose basically most of the mainstream amplification technique. I think most of them will work pretty well in Magnify specimen.
Okay, great. Thank you so much. And thank you everyone who submitted questions. If we did not get to your question live, I will have a slide at the very end that will include Dr. Zhao's email, so you can email him directly with your questions. I will go ahead and share a couple slides. All right, so with the last few minutes, I just wanted to share a few product slides that might be interesting and relevant for our audience today. So Proteintech does offer several solutions for our customers that are doing IF and microscopy. So in addition to the thousands of primary antibodies that we offer, Proteintech also has 2,800 antibodies that are conjugated for IF. These antibodies are directly conjugated to various Coralite plus dyes, making them ideal for multiplex experiments. And all our Coralite conjugated antibodies undergo very stringent validation so that we ensure that there's low background. They also have unmatched brightness and photostability, and they're similar to Alexa Fluor in spectral profile. And like with other conjugated antibodies, they're really the best choice for high abundant targets. Proteintech also understands that, a conjugated antibody for your target of interest might not always be available. So we've made a solution for that too. So Flexible is a novel antibody labeling kit that uses an affinity linker to conjugate fluorochromes, enzymes, and molecules in any buffer condition in just 10 minutes. So this means that you can label your antibodies for IF in your lab and eliminate that secondary antibody incubation step. And because the antibodies are labeled prior to incubation with your sample, Flexible is suitable for same species multiplexing. So you don't have to worry anymore if that your favorite antibodies are all from the same species. You can basically just label them and use them in the same experiment. Sometimes the traditional primary secondary approach is just what's ideal for certain experiments. And so Proteintech also does offer secondary antibodies for this method of IF. So here we're highlighting our nanosecondary reagents, which are nanobodies that are conjugated to Alexa Fluor or Coralite dyes. Our nanosecondaries are recombinantly manufactured and now have a defined degree of labeling. So that's really going to be ideal for the reproducibility across your experiments. They also have very high affinity for binding to primary IgG antibodies in a subclass specific manner. And so this is going to result in cleaner images. So you can see an example of this here on the right, where you can see that each nanosecondary that was tested only recognizes one subclass of IgG compared to the traditional rabbit secondary that you can see here at the very bottom that we obtained from a competitor, which recognizes the rabbit IgG, but also some other subclasses of mouse IgGs. Also, since nanobodies are so small, because they're about 10 times smaller than traditional antibodies, they have the ability to penetrate tissue better and bind very closely to your protein of interest. So this is going to be great for generating high-res images using, confocal or super resolution microscopy, which you can see here in this image in the middle, that where it was imaged, the vimentin was imaged in HeLa cells. And then finally, I just want to highlight a product that we've more recently added to our portfolio, which is our multi-Rab recombinant secondary antibodies. So multi-Rab is quite unique when it comes to secondaries because it has a high sensitivity. It has the high sensitivity of a polyclonal with the superior sensitivity of monoclonals. And the way we did this is by precisely engineering a mixture of recombinant monoclonal antibodies that were carefully selected based off of their performance. Because the mixture is made up of multiple antibodies, they recognize multiple complementary epitopes on the same IgG. Multi-Rabs also have very minimal cross-reactivity with off-target species, and since they are recombinant, they have an unlimited supply, high lot-to-lot consistency, and reproducibility. So this is really going to be perfect for kind of your long-term IF projects. Currently, we have multi-Rabs that are specific for rabbit and mouse in the various conjugates that you see here on the right. So I just wanted to end with this. As an attendee of today's webinar, you will receive a follow-up email that will consist of a recording of this event. You will also receive a certificate of attendance as well as a link to request a 20 microliter free multi-Rab sample. If you're interested, you could scan the QR code right now and request it, but you will also receive a link in a follow-up email. And please do allow 1 week for the follow-up email to be sent. So with that, I just want to thank everyone for joining. If after I end the webinar, you do have any questions, please feel free to email us at events@ptglab.com. Or as I mentioned, if we did not get to your question live and you have any further questions for Dr. Zhao, please do feel free to email him as well. And if you liked this workshop and you want to know about any of our upcoming events, please do follow us on our social media channels because we do post about all our upcoming webinars on there. So with that, I would just like to thank everyone once again for attending. Thank you so much, Dr. Zhao, for that really informative talk and all the beautiful images that we got to see. So I hope everyone has a great rest of their day. Take care.
Thank you so much for invitation and all the interest. Thank you.